Welcome to Professor SNAB, where we aim to make A-level biology as simple as a cell. So today we're looking at a core practical, where we will be carrying out the study on the ecology of a habitat, such as using quadrats and transects to determine distribution and abundance of organisms. And we will be measuring abiotic factors in the habitat to do so. So, a quick introduction into the practical. So, the distribution of the species is determined by abiotic factors. So, these and biotic factors. And abiotic are the non-living components of an environment, a habitat, and biotic factors are the living components of a habitat. And examples of abiotic factors are light intensity, amount of water and nutrients available, temperature, and soil. And some examples of biotic factors are predators, prey, disease, parasites, and competition for resources. So if you want a more detailed video focusing on abiotic and biotic factors, there is one uploaded before, so you can have a look at that. And let's move on. So the apparatus that you need for this practical, so the first thing is obviously a quadrat. Which will be used to measure the distribution of the biotic or biotic factor you want to measure. Mostly biotic if you're using a quadrat. And same for transect, another possible tool that you can use to measure the biotic factors in your environment. You also need a clipboard to take notes on your practical. And finally, you need certain equipment depending on what you actually measure. So if you're measuring light intensity, you would need the tools to measure light. And if you want to measure the moisture of soil, then you need a completely different tool for that. So that depends on the experiment you want to carry out. So what is the method? So you first find a site where there's a gradual change in your chosen abiotic variable so that you can actually observe the change. So when you do find a place the transect down and choose a species that changes in abundance along the transect. So these can be plants or these can be insects, a certain type of flower and whatnot. So place the quadrat at each of the marks on the transect, placing the bottom left corner on the mark every time. And every time you place the quadrat, record the percentage cover for the chosen species. So how do you find the percentage cover? A percentage cover is calculated by counting the number of squares a species is part of and dividing that by the total number of squares in the quadrat and then timesing that by 100. And squares must be counted even if half or more of the square is covered. But if it's less than half, then you can ignore it. At each coordinate, a measure of the independent variable should be taken. For example, if investigating light intensity, a photometer can be used to take the reading for the light intensity at each coordinate. So every time you stop, for example, if we assume you're measuring the relationship between a certain plant species and light intensity, so every time you put down your quadrat on the site, you also need to measure the light intensity at that site. So what are the risk assessments for this practical? So there's the obvious one, biohazard, because allergies are possible because of soil bacteria. Contamination is also possible during the practical. So it is safe to wash hands after the practical to minimize the risk. And if you're someone that has hay fever, then it is safe to carry hay fever medication in case a reaction is triggered. So in case of an emergency, then do seek assistance. So another risk assessment is slippery surfaces. So ensure appropriate footwear is worn during the practical And in case of slips and falls, seek appropriate medical attention. So another important point is after the practical, what you would need to do is to plot the data. So the independent variable, which would be the usually the abiotic factor, for example, in our example, it would be the light intensity, and this would fall on the x-axis because it is the independent variable, and the percentage cover of the species you are measuring would fall on the y-axis because it is the dependent variable, and this depends on the independent variable that you're measuring, in this case, the light intensity. And once you plot the data, then you can carry out different statistical tests such as Spearman's rank, t-test, or chi-square. So final conclusion, the plotted data will show you correlation between an abiotic variable and a biotic variable. So for example, you can expect in our example that with an increase in light intensity, there's an increase in the number of plant species that you can record from your quadrat, or the other way around. A decrease in light intensity means that you have less number of plant species recorded in your recorded by a quadrat. So that's kind of the expected data that you will see, the expected trend that you will see. However, it must be noted that correlation is not causation, and multiple factors could be influencing the result. And this kind of remembering this point can help you develop really good analysis in your report and actually look deeper into possible other influences that can have that must have resulted in the data you've collected. So that is all from Professor Snap today. Hope this has been useful. If you have any questions, please do drop in the comments below, and don't forget to like, share, and subscribe. Thank you.